That's right, that's right.
Yeah.
Small plastic. General one.
571 or something.
567.
I might have sat in a class like that. You didn't overlap it.
I did. How about you?
Pardon me? I'm longer next to having a class.
Oh boy.
Brutal. I've been doing some very basic economics research. Contest theory is applied to scientific grounds.
Right, we've had that conversation several times. Do we need a bigger room?
All right, welcome. It's noon, so we're going to get started. My name is Jason Delborn. I'm a faculty member in Forestry and Environmental Resources and also one of the GES cluster faculty. See some new faces in the room, so thank you so much for coming. We run this colloquium every week with speakers from NC State as well as from outside. We're really fortunate to have this kind of event that brings our community together for these very interdisciplinary discussions on a regular basis. We always start just by announcing any upcoming events that are related to GES. One of them is, one of the students I work with, Jessica Barnes, is defending her dissertation on Thursday morning at 9 a.m. in Jordan 1214. And she has, her work focuses on the genetically modified American chestnut, and it's a very diverse dissertation. She does some modeling around biogeography of the chestnut and where it might end up if it's restored. She questions whether biotechnology is inherently neoliberal, and she also applies tools of anticipatory governance to thinking about the way that restoration could happen in a more responsible way. So she's one of those students that really has embraced the diversity of disciplinary and methodological approaches in GES. So I encourage you to come and listen. The first part is open to the public, and then we have a closed session. What other events are coming up that we need to be aware of? Right.
Yeah, so just to restate that we're going to have a symposium June 4th and 5th. That's going to be both a capstone of the IGERT as well as bringing in people from outside to discuss the future of integrative graduate education. And we have two keynote speakers. One is Terry Lomax, who used to be in charge of the ORIED program and then a major position at the RTI Institute, who will be talk about what workforce needs in terms of interdisciplinarity, and then the director of the NSF National Research Traineeship Program is going to be the other keynote speaker, and we have others coming in. So June 4th and 5th, you'll be getting invitations. There's a website. We just have a couple of little things to do, so you will be hearing about that soon.
Thank you, Fred. Yes, sir.
There's a seminar tomorrow in crop science by Wendy, see if I can pronounce it, Srinik from Corteva. She'll talk about CRISPR in agriculture, and that's at 3:30 in Williams Hall.
Thank you. Any others? Okay, so just as a preview, next week we have a speaker. He's a scientist who's been working on the genetically engineered American chestnut tree up at SUNY ESF. He's a relatively junior scientist. I think he's still a PhD student, but he's been working on this project for quite a long time, and he's been quite active in preparing the regulatory materials for that go to the EPA and USDA and FDA around the chestnut tree. So he's going to be giving a talk here on Tuesday and then attending our stakeholders workshop later in the week. So please come back next week to hear him. So I'm going to have Fred introduce our speaker today. So come on up.
Okay, so some of you were at Bruce's talk yesterday, so you already heard my general introduction. But just to say pretty briefly that Bruce is probably the most eminent researcher doing work on resistance management in the world. So that's who we have with us today. But I just want to say that beyond that, I mean, you know, he's had over 300 papers published, and 27 of them, I think it was, maybe it's 28 by now, were in PNAS or Science or Nature, right? So I don't have to give him his credentials here. He's a Regents professor at the University of Arizona. But Bruce is also, you know, a very important person in terms of these issues beyond the research, in terms of getting things done, in terms of resistance management, how to take it out of the lab and actually have it happen. And he's been fighting the good fight in some ways for a long time now. And there are rough waters sometimes. And we talk a lot here about the book on, you know, being an honest broker and so on. But he's going to give us, you know, basically his sort of view from the field in a certain sense. But Bruce has also been very instrumental in other ways in terms of making community. He was the Department Head of Entomology in University of Hawaii, and has now, I think it's almost 2 decades that you've been the Department Head at University of Arizona in Tucson in the Department of Entomology. And there too, there's also being an honest broker in terms of keeping your faculty in a positive mode moving forward, and he's done a marvelous job in doing that. So I'll let Bruce take over, and Bruce is going to talk to us for 20 to 30 minutes, and I think he's going to be kept on time here,
Yes,
and then we'll have some discussion.
Well, I'm going to have to talk fast, but I know that the discussion is going to be great, so I'll try to be as brief as I can and give you some key information material for the discussion to follow. So, challenges of trying to be an honest broker on insect resistance to genetically engineered crops is my title. And as you know, there's a chasm between the two sides in this debate, and it's been very polarized with very little middle ground. And as part of being an honest broker, or trying to be, the first thing I have to be is honest with you about my conflicts of interest, and I've got plenty. Most of mine fall on the pro-GMO side. I've been funded by the major companies that produce and sell biotech crops. And also, I put USDA here on the pro-GMO side, and Cotton Incorporated as well. And I've been received and continue to receive research funding, and I've also done some consulting. But I'm kind of eclectic. I've also been supported from anti-GMO forces, if you will. AMVAC is a company that sells insecticides, and so they compete with GMOs in my realm, and so they're very happy to hear news that there's insect resistance to the GMOs, because then they can sell more insecticide. And the Union of Concerned Scientists has taken an anti-GMO stance in the agricultural realm. Just a little bit of background biology. Bacillus thuringiensis, or BT for short, is a natural widespread bacterial pathogen of many insects. It's been around for forever, more or less, for millennia. And it's been known and used for more than a century to control insect pests. And the really cool thing about it is that the proteins that this bacteria makes that kill insects are very specific. So you can use them and kill, certain ones will kill certain pests and not harm other organisms, and in particular, not people and even natural enemies, that is, other insects, good insects that eat or otherwise destroy the pests. And the genes from the bacteria from BT have been engineered into crops. This happened about 30 years ago. And the proteins I'm going to talk about are mostly crystalline proteins, and they're called cry proteins for short. So the benefits of these genetically engineered Bt crops are that they are highly effective against certain target insect pests, and they can even kill pests that bore inside plant parts. Those of you who heard my talk yesterday, please forgive me, but it's not that these pests, by boring I don't mean dull and annoying, although they may well be that. By boring I mean they're inside the plants and they're essentially impervious to insecticides. So this has a potential benefit, in fact has been realized for many of these crops, but they reduce use of insecticide sprays, which is a good thing. So these can have economic, environmental, and health benefits. Because of these benefits, this technology has been adopted at an incredibly rapid rate, one of the most rapid rates of technology adoption in the history of agriculture. So in 1996, the first year these crops were grown commercially, there were about 1 million hectares grown worldwide, and last year close to 100 million hectares of Bt crops grown worldwide, including a very high proportion of corn and cotton in the United States, and a high proportion of soy in Brazil. In 2016, 20 million hectares grown there. And so one thing we know as entomologists, and it was recognized by Rachel Carson more than 50 years ago in Silent Spring, is that insects are very good at evolving resistance to insecticides. And this shows the number of insect species with resistance to one or more insecticide. Around 1960, Silent Spring, we were about 100 species, now over 500. That's kind of leveling off, but if we think of cases of resistance, so not just the species, but we say the species, the insecticide, and the location, that's actually going up faster now than before, and we're over 10,000. So early on, leading experts, including one in the room, had the vision to see the potential problem of evolution of resistance to these Bt crops. And Fred did some modeling that showed under the worst case scenario, we could see resistance to Bt crops in as few as 3 years. So I'm going to give you a quick review of what actually happened over the last 2 decades. Here's the first 5 years, all the cases of field-evolved resistance to Bt crops worldwide, there were none. Zip. So that worst-case scenario did not come to pass. But if we go now out to 10 years, there was only one pest species, it's called Pasea. It was the first insect to have documented resistance to a Bt crop. It was resistant to 3 different Bt proteins produced by Bt corn and Bt cotton. It was very controversial, and I'm going to tell you more about that later. So we'll come back to it. But by the end of the second decade, that is 2016, the picture changed dramatically. And by then there were 7 different insect pests with resistance resulting in a total of 18 cases worldwide. Most of those are lepidopteran pests, caterpillars, 6 of the 7, but one of the 7 we call the billion-dollar beetle. That's the corn rootworm. It's now resistant. It's this one here. It's now resistant to all 4 Bt proteins that have been deployed commercially in Bt corn to control it. So what the question I've been addressing with my team for the last few decades is, can we apply evolutionary principles and knowledge about resistance to make Bt crops more sustainable? I think the answer is yes, and I hope, well, I won't have the time to show you the detail today, but I think we've demonstrated that. We've learned that, as I just showed you, field-evolved resistance to Bt crops, there were only 3 cases by 2005, but 18 by 2016. And resistance to Bt crops is actually evolving faster now than initially. Part of this is because of cross-resistance. That is, a pest becomes resistant to one Bt protein, and that allows it to be resistant to another related Bt protein. And consistent with evolutionary theory, and predictions that were made before we knew what the patterns were in the field, resistance has evolved slower with recessive inheritance of resistance in the pest, low initial resistance frequency, and the parameter that's most relevant for the discussion today is abundant refuges of non-BT host plants. So giving those pests a portion of their habitat where the susceptible individuals can survive and thrive, so that the entire population is not being selected for resistance. One of the strategies is to make BT crops now that produce 2 or more toxins that kill the same pest. That can be helpful in certain situations, but it's by no means a panacea. Whether we have single or multi-toxin BT crops, and these conditions are not met, if we don't have recessive inheritance or we don't have a low initial frequency, we can counter that. The evidence says that if we provide large refuges, a high proportion of the habitat where the plants that the insect feeds on are not producing the BT proteins, then we can delay resistance. And that means we use the BT crops in combination with other controlled tactics in integrated pest management, or IPM. So the goal of resistance management is to delay evolution of resistance in pests, and this works best when it's done proactively. So the sooner we put the tactics into place, the better. And resistance management for BT crops in the United States has been regulated by the EPA from the get-go. Why? The rationale is that BT crops can, in fact, they have decreased insecticide sprays when they work, because you have a different way of controlling the pest. So this provides environmental benefits. So it's in effect, the susceptibility of the pest to the BT proteins is a public good. So the EPA wants to avoid resistance or delay resistance to conserve the environmental benefits of the BT crops. How is this regulation carried out? When a company wants to sell the seeds for the BT crops, they have to register these crops with the EPA, and the EPA requires a resistance management plan. That includes tactics. The main tactics is refuges of non-BT host plants. And so the key variable there is what's the percentage of the host plant, let's say cotton. What percentage of cotton does a farmer need to set aside as a non-BT host plant refuge? And the second sort of twist on that is how many different toxins are targeting a particular pest. So if you have a single toxin targeting a given pest, you need a larger refuge to get the same amount of delay and resistance in principle than if you had multiple toxins. These plans also include resistance monitoring. That's a requirement for all the BT crops in the United States. And there are also remedial action plans. So say you detect some resistance, what are you going to do about it? So if we're going to do anything useful, we have to have a definition of resistance. I'll call it the academic definition. It started actually with something that came out of a U.S. National Research Council study in 1986. Fred was one of the organizers of that, and this definition has evolved a little bit over the years, but it's for field-evolved or field-selected resistance is a genetically based decrease in susceptibility of an insect population to a toxin that's caused by exposure to a toxin in the field. So the key point here, or key point, genetic change. And this is a proactive definition. So early detection of resistance, a relatively small change, can trigger some kind of response, some kind of change in what's required or what farmers do to delay the resistance at that point. And I think it's objective. So we can do experiments. We can find out if there's a genetically based decrease in susceptibility or not. We can answer that yes or no, basically. And based on the history of the situation, we can say whether it's been caused by exposure to a toxin in the field. In contrast to laboratory selected resistance. I'm going to contrast that with the industry definition of resistance that they still use now. This is from Iraq It's a global organization, Insecticide Resistance Action Committee. This group consists of the biotechnology companies and other companies that sell insecticidal products. So they say it's a heritable change in sensitivity of a pest population reflected in the repeated failure of a product to achieve the expected level of control. So we start off the same, it's a genetic change, genetically based decrease in susceptibility. That's the common ground. Here's where it differs. This is a reactive definition. According to the industry, we don't have resistance until we've had repeated failures. But by the time you've had repeated failures because of resistance, it's too late to do anything about it. So you can't, by this definition, you can't have an early response because you don't have resistance until it's all over. I consider it subjective because it says repeated failure to achieve the expected level of control. Well, the expected level of control is not defined. So you could say, well, I don't expect this product to work very well. If it doesn't, then it hasn't failed. So anyway, I don't like this definition very much. Long story short, we kind of have a war of words in part about the definition. And for a long time, it wasn't very productive. It was like, okay, here's the data. And I and some people working with me would say, well, that's resistance. And the industry people would say, no, it's not. And yes, it is. No, it's not. We went back and forth like that for years, actually. It's easy to see in hindsight. And one recent paper, actually by a graduate of NCSU, illustrious former student of Fred's, said that the problem with this proactive definition is that it could trigger unnecessary resistance remediation. But the actual fact is no cases of field-evolved resistance have triggered regulatory remediation whatsoever. Ironically, as the cases of resistance have increased, the EPA has actually reduced refuge requirements. And this is in part because now we've moved from plants that each produce one toxin to plants that almost uniformly produce 2 or more toxins targeting each pest. So trying to find common ground and avoid this back and forth yes-no argument. On my side, we recognize and have published and explicitly state that the field-evolved resistance is not all or none. So the impact of field-evolved resistance depends on the frequency and the magnitude of the resistance, the spatial distribution, the pest population density, and the availability of alternative controls. That was kind of step one. And step two was saying, all right, we're going to provide categories of field-evolved resistance. So field-evolved resistance is still a significant genetically based decrease in susceptibility, but we define this red category, practical resistance, because it's resistance that has practical consequences for pest management. So it's not some tiny increase that's statistically significant, but maybe would never make a difference to a farmer. And we have objective criteria, so you have to have some experimental evidence that there's at least 50% or more individuals in a population are resistant, and that reduced efficacy of the product is reported from the field. And all the other cases that are field-evolved resistance but don't meet those more stringent criteria, we put in the yellow zone, caution. We call that early warning. And if there's no significant decrease in susceptibility, that's the easy one to agree on. They're susceptible, the green. And we've recently published a review of essentially all the monitoring data worldwide on this topic. And I updated it just a little while ago. So there are now 38 cases where there's well documented data on what's happened in the field. And here's how they break down. 18 in the red zone, practical resistance, 3 the early warning, and 17 cases where the pest is still susceptible. And you can see the worldwide distribution. So in response to this paper, here's a little a quote that was included in a press release from the University of Arizona about the paper. The point here is that we always expect resistance to evolve, and those 17 cases still in the green zone, some of those have been 2 decades or more. So I said, if we can delay resistance from a few years to a few decades, that's a big win. Here's what an anti-GMO person, Liz O'Neill, said. So she says, and I say, if in some cases the measures delay development of resistance by a few years, that's a big win. And she goes on to say that tells us all we need to know about the GM industry's vision for the future of farming biodiversity in our natural environment. Well, that's criticism from one side of the debate. Now I'm going to focus on this controversial case, Hilgever Pasea, the first case of field-evolved resistance to a Bt crop. There was some landmark work done, just a classic study by a group led by Randy Luttrell, who was at the time at the University of Arkansas. And they did exactly what you're supposed to do to track these changes, to determine if resistance has evolved. Before Bt cotton was commercialized in 1992 and 93, they evaluated the pest response to the Bt toxin, in this case Cry1A-C, that was going to be available in the commercially used Bt cotton. And then they did the same study after to see what if any changes had occurred. And this involves laboratory bioassays. Essentially, insects are collected from the field. The progeny are reared in the lab under standardized conditions. And then the larvae are tested on food treated with the Bt toxin. That's what was done in this case. Sometimes people use the Bt plant material itself. But here's your insect chow, and you can treat it with a series of concentrations of the Bt toxin and see what kind of mortality response you get. So here are the results. I want to define one of the parameters that's used to track resistance. It's called the resistance ratio. So the LC50 is the concentration killing 50% of the population tested. And the resistance ratio is the LC50 of a field-derived strain divided by your standard susceptible laboratory strain. So the higher the resistance ratio, the more confident you are that there's resistance. And this is actually a conservative method of evaluating resistance, because you're looking for changes in the midpoint, the median of the population. This is Randy. Here are the initial data, and they look like what you'd expect. So the maximum resistance ratio, take the highest LC50 from all the field populations divided by the susceptible lab strain, the ratio was 1.2. So basically there was no difference between, actually overall they had several susceptible strains from the lab, several field strains. There was no statistically significant difference. You can see they're all the points are crunched down here in the susceptible end. We have the LC50 on the x-axis and the proportion of strains falling into a category on the y-axis. So fast forward, this is before Bt cotton. A decade later, this is now after 6 to 8 years of Bt cotton, we still have a high proportion of the strains, about 75% are still susceptible. They're still on this end of the distribution. But now we have lots of resistant strains, and I've highlighted the 1, 2, 3, 4, 5, 6 most resistant strains. So this one out here with 578 for a resistance ratio, that means it took 578 times more Bt toxin to kill 50% of that strain compared to what it would take to kill 50% of the standard lab susceptible strain. That's a really big change. Very strong evidence of resistance, and particularly because it's not just one strain. There's 2 over 500, there's 4 over 100, and the 6 with 50 or higher. So that's very compelling evidence. It turns out that before they published the journal article about this, they published a proceedings paper that actually had more information. It was published in 2004, and they concluded the LC50s were higher than those measured before commercial release of Bt cotton and Bt corn. Very straightforward, yes. That's what the data show. They even did crosses and tests of inheritance, And the resistance, their work showed, was not recessive. So this is bad news for resistance management, but it also fits with the idea that this pest evolved resistance readily. Its resistance was inherited as a dominant or incompletely dominant trait. That favors faster evolution of resistance, so it's kind of consistent with the evolutionary framework, the predictions about resistance. They also reported, in addition to their diet bioassays, that's treating the caterpillar chow with the Bt toxin, the more resistant colonies also had higher survival on Bt cotton. They did experiments to show that. And finally, two of the resistant strains that they established in 2002 were collected from Bt cotton fields that had unacceptable levels of boll damage. That's the cotton bolls. So this fits the definition of practical resistance. It's not just some statistical thing. It's not something just cooked up in the laboratory. All the elements are there. So I'm not really sure exactly what happened. I've been talking to these people these many years, but apparently it had a change of heart between publication of the proceedings paper, which was titled Resistance to Bt in Arkansas Populations of Cotton Bollworm. Here they made the very straightforward conclusion from the data that LC50s were higher after Bt cotton than before. When they published the journal article in the Journal of Economic Entomology 2 years later, the title was changed to Susceptibilities to Bt, from resistant to susceptibilities. And mostly they avoided the question about resistance altogether, but the one sentence that's kind of analogous to this one, now they say Helicobert Pasea populations may have higher LC50 values, may have. Well, they were higher. You can do a statistical test, do whatever you want. They were obviously higher, but somehow their interpretation was changed. And I was in the position of reviewing the data on this question for a book chapter, and I came across both the journal article and eventually the proceedings paper, and decided that this was big news. It belonged in a forum that would be more widely read than a book chapter, and drafted a manuscript with one of the main conclusions that this pest, Helicobert Pasea, had indeed evolved resistance to Bt crops. But we wanted to be broader and more useful than that, so we modeled resistance evolution in 5 major target pests. We compared the predictions from modeling to what had actually happened in the field. So here we have the percentage of refuge and then the years predicted for resistance to evolve. This is the pest, Helicobert Pasea, that evolved resistance first. And the modeling suggested it's because of the dominant inheritance of resistance that this pest, for any given refuge percentage, would evolve resistance faster than all the other pests that were major targets of Bt crops at that time. So I shared this with one of my colleagues, someone I'd known, co-authored papers with at that point for over a decade, a Bt expert. He was at Auburn at the time, and this is what he emailed me back. And this was kind of my wake-up call to a world that I never imagined. But he said, whether we have field-evolved resistance or not, your statement that we do would be devastating. Why? Your statement from a highly successful scientist, I like that part, that works, would be all the ammunition many special interest groups would need to persuade removal of Bt cotton, non-adoption of GM crops in countries that desperately need them. So this line of reasoning is basically, the guys on the other side don't play fair, so don't give them any ammunition. They're already anti-GM. Don't give them actual facts that they could use to convince people that this technology should not be used more widely. So I was pretty stunned by this. Eventually, we went on to publish the paper 2008. Monsanto immediately came out with counterclaims, and my former collaborator and former friend was the lead author on the letter to the editor. They also had several other friends and former collaborators who emphatically disagreed with our conclusions and didn't mention anything about the rationale that was presented in the email, which is, as far as I understand it, was the true rationale for disputing these claims. But they had like a series of pseudo-scientific reasons, like, well, those diet bioassays don't mean anything, that kind of stuff. Anyway, we also published rebuttal, point-by-point rebuttal. And I can tell you there's some satisfaction in the citation count. History has sort of been in our favor. The paper has been cited nearly 500 times, the letter about 1/10 that. But the controversy continues, and people who want to cling to the claim that this insect is not resistant, they mention this letter, and they still cite some of the same reasons even now. I'm going to have to, I'm going to actually skip this. Well, I'll tell you that a paper in PNAS just came out showing that BT corn has regional benefits in reducing the abundance of this same pest over time, and this is in the mid-Atlantic region. And this kind of counters, I guess what I think is true, that the pest had evolved resistance. I'll back up for a sec, let's see if I can. Yeah, there was additional data came later showing that this insect was also resistant to Cry1AB, which is produced by BT corn. But it turns out that these data are pooled for 3 states. That's where the benefits were realized. But most of the data come from 2 of the states where apparently resistance has not happened. So resistance is not necessarily geographically uniform. And if just before coming, I looked at Maryland alone, where the resistance had been documented. And here we see that the abundance of this insect, we got the log of the number of the moths trapped per night over time. You can see it went down like a stone until 2006, but when I think one explanation is when resistance kicked in from thereafter, the abundance has not declined at all. So that's probably why there isn't a discrepancy when we break it down to the area where resistance evolved, rather than pooling 3 states, some of which may include many populations that are not resistant. So I'm going to, an update. The EPA is convening a scientific advisory panel in July to look at this question of lepidopteran resistance to BT corn, actually, and BT cotton. And I'm going to, all right, I'll tell you. So this reminds me of when I was in Yellowstone a few years ago. This controversy, for most of the time, it looms kind of in the background, doesn't bother me that much. After all, I've got tenure, I've got nice friends, I got a nice home. But here, sort of vaguely in the distance, there's this grizzly bear. But in Yellowstone, I'm standing on this boardwalk. It's elevated. I'm safe. The bear's off. In the distance, kind of like this controversy, doesn't really affect me day to day. However, everything changes when the bear decides that I'm dinner and it wants to come on the boardwalk. So once in a while we get bitten by this controversy, if you will. And here's an example. This is an anonymous review excerpt criticizing a proposal we submitted to the USDA, which was, in fact was rejected. And it says reports published constantly. Okay, we like that. By these researchers are often used to misinform the general public and fuel the science-lacking arguments against agricultural biotechnology. So I'll stop there and open it up to your questions. Dominic.
Great seminar. It was really interesting to hear you give the dialogue from those papers, haven't read them, so thanks for that. I want to go back to the beginning, to your definition of resistance and how many cases there are. If you if you talk to the EPA, they have these regulatory triggers to trigger regulatory action if resistance is reached, and they said they've never once had it reach the regulatory trigger for resistance. So we have all these published papers saying they're resistant, but we're not hitting the regulatory trigger for the EPA. I mean, other than like the scientific advisory panel, what's the solution? Or for finding some middle ground there?
So you mean, why is the EPA so slow to respond? Why aren't they doing what they more or less said they would? Is that?
Well, from my perception, when I talk to the companies and you talk to the data that's required for them to submit to the EPA to hit that regulatory trigger, I think they're doing their due diligence. They're submitting the data and it's not hitting the regulatory trigger, but what's the problem? Do we need to change that regulatory trigger? Do we need to change how the data are submitted? You know, what's, in your opinion, what's the answer to that?
Well, I guess you're saying it's a problem that the EPA doesn't respond, right?
I think so.
Yeah. So I think that's a more of a political problem. And so I had left this out, but so when they're calling this for the scientific advisory panel, they are including Helicobacter Pasea, but they say data were published in 2016 showing resistance. Well, the first data showing resistance were published in 2004. So I think there's some kind of political, social reason for the EPA not responding. It's not. You could have a better, more precise decision of resistance, but that would kind of beg the question: would they would they actually act? I think this ambiguity about the definition, even though the EPA considers it a problem, it's actually allowed them to not act. And in some ways, that's politically favorable for them.
So, just a comment with respect to that, because one of the ways that, in fact, the majority of the ways resistance issues that raise to the level of a company going and investigating are reports of some issue with failure or performance. Well, that issue of performance is highly, as you say, subjective. It's very problematic because it's not just what you expect. It's, as you indicated, the infestation level that you have. So you can think of if you have 95% or 95% efficacy against a population that's like 10 insects per plant, you get less than one per plant, right? But if you end up with a situation like you would with some insects of 50 per plant or 100 per plant, you have a control failure. And in some of these things, corn rootworm being one of them, those populations fluctuate dramatically from year to year. So you get a report of a problem at this location this year, and then the problem goes away the following year, and in large part, or at least to some extent, because you just don't have an infestation and you don't see it. So the criteria that you set as an objective criteria in an assay of whatever standard, this proportion of individuals tested show up above a threshold you can relate to that. But that also requires that you go out and you monitor all the populations. So if you're not doing that kind of labor-intensive monitoring, you fail to detect these problems.
But as Dominic was saying, so the companies that sell these products are required to report the monitoring data every year. So the EPA has the essentially raw data showing what the responses of these populations are. So they do have that information. Then it's a question of interpretation.
Yeah, so one of the most interesting things that I observed while studying this phenomena of many different stakeholders from many different backgrounds making an argument is they have their argument, they have their own data, they have their own fact. But when something happens, no one takes responsibility. So is there any way that there's some system that actually actually push some responsibility to certain part, so people can think twice before they say all right?
Good question.
I wonder if you could talk about across these different species that are targeted by BT, sort of what alternative approaches there are for management, and how does options for alternative management strategies change the dialogue about the sense of urgency, essentially, for managing resistance. So I think about examples from, sort of before I was in science, but what stories I've heard about pests evolving high levels of resistance that then couldn't be managed by anything else. And that generated a sense of urgency to do something about managing resistance to upcoming technologies. So I guess I wonder, across these different species, are there examples where there is indeed that sense of urgency, and I guess how that might change the dialogue about this going forward?
Yeah, it varies, I guess, across the species. So the corn rootworm, the billion-dollar beetle, crop rotation is a good strategy in many areas, not all, because this pest has actually evolved resistance to crop rotation in some places. But that's an example, but it's not as palatable, it's not as easy for growers, and so it's not readily accepted, but that's like the #1 backup for that pest. And in fact, rather than the backup, it should be used in jointly with BT crops to make them more sustainable. I guess Helicobacter Pasea is the one I'm most familiar with. The alternative is insecticides, sprays, and they don't work all that well. It was on one of the slides, but in a part of Maryland where the BT resistance has been detected, growers are spraying their sweet corn up to 12 times a year now. So the alternatives are there, but they're not as desirable. They're more expensive, less environmentally favorable.
So you gave great examples of some scientists feeling the need to question anything that goes against their desired narrative for original plants, and how that leads to real inability to deal with nuance around findings, or and really just have any sort of nuance around this issue whatsoever. And given your experience in this realm, I'm just curious what you think needs to be done to address that issue.
Need a truth commission. No, it's a good question. I mean, putting it in the best light, I guess you could say there are people who believe that something is good for, like for the world. So they should do, the outcome trumps their responsibility to be honest and to be scientific about something. Because it's more important that, let's just say, for example, to feed the starving children of the world than to be honest about whether there's resistance to BT or not. So it's the ends justifying the means. And I don't know what to do about that other than fight back, speak up. But I'd be interested to hear what other people have to say.
So if you were at your red resistance level, like right at 50%, and it was dominant inheritance, then about 17% of the population doesn't have a susceptible allele. Is there any idea if at that or any other point you were to withdraw the selection, whether you could drift back to full susceptibility, or whether you're stuck with rapidly evolving populations from then on?
By the time you get that high, it's really difficult to restore susceptibility. I told a story yesterday where in China, resistance was detected initially, but the average percentage of resistant individuals was only about 2.5%. And it was a recessive trait, and it was actually reversed in the field by reducing selection, by planting a higher percentage of refuge, cotton in this case. And so when it's at a low percentage, yes, but once it gets to that high a level, it's very difficult. And it actually it takes more than just withdrawing the selection. You have to have selection acting against the resistance. But that is often the case. There's often a fitness cost associated with resistance. So you can go backwards, but generally the selection going backward is much less intense, less strong than the selection favoring the resistance. And you don't go back to the complete susceptibility, because it's very hard to drive the population, say, below a few percent resistant.
So how do you feel when your results are used by critics of GMOs? And how has that experience changed the way you communicate your science, either formally or informally?
I guess I'll just, I'm supposed to be I'm trying to be an honest broker, so I'll be honest and say that I prefer to get attention than to be ignored, so I'm not terribly bothered. On the other hand, I guess it makes me realize, like, what some of my former friends and former collaborators are nervous about, so I try to be super careful and not say that could be misinterpreted. So I try really hard to be clear, try to be objective and be clear, so that I think of people who are hostile to whatever it is I'm saying, either on one side or the other, and so I feel comfortable that what I said is accurate, it's as objective as I can possibly be, it's backed up by evidence. And then if they, people have their biases and they want to, this means the sky is falling, this means we've solved all the problems of the world, if they want to say that, then that's their responsibility.
I guess I'm wondering, like, it just seems like both sides of this debate have a profound misunderstanding about the implications of resistance for their respective stakes in the matter. So if you talk about the pro-GM camp, it would seem like they would have a major interest in managing resistance in order to sustain the effects of this technology. And conversely, like the anti-GM people would almost want to accelerate resistance to abandon the technology as quickly as possible. So I'm just wondering, like, particularly the scientists, like the reviewers, why they don't, in your interactions and discussions, do you sense that they just don't get, they're not thinking in the long term or something?
No, they actually, industry in particular, and a lot of the people who are pro-GM give lip service to sustainability, and they call it stewardship, product stewardship. These are catchphrases, and all the, they have resistance management plans. It just, well, I guess it's partly the people I interact with are the scientists, and there's actually a fair bit of common ground, but there are other people in these companies, and they seem to have sway, actually. They're in marketing and sales, and they don't want anything said or done that's going to reduce the sale of their products. So, for example,
this year.
That's right. This year, they want the highest market share possible this year. And at one point, after we published that first review, that was in 2008, Monsanto sent someone to speak to me, and he said, we know that you're a good guy, we know you understand all this, but other people don't, and we can trust you, but we can't trust them. So we don't want to see resistance and one of our products mentioned in the same sentence.
I've also got a question, I think that builds on the last couple. It seems like your stories and the company scientists you just described about the future are really different. It seems like you have really different expectations or maybe even hopes for that future. So I guess the company scientists you described seem to assume that some other product will come along or some other trait can be stacked so that the pesticide can be saved or they'll move on to another product. But you seem really committed to this toxin. You've got a thing for BT. And I wonder if you can tell us about that. I mean, is that because you start your story 100 years ago and you think this is a really important tool? Or is why is it that you love BT so much and you think it should still be available as a tool in 20 or 50 years?
Well, maybe because it's my initials.
Did you change your name?
Yeah. So I think I think there's a continuum. So I love BT, but I'm also happy that there are some new things in the pipeline that are similar. But the problem is it's not so easy to find proteins or other ways of controlling insects that are as good as BT. There are new things being found, and some of them look really promising. And there are hundreds. It's not like there's just one BT protein. There are hundreds of them, and there are also ways of modifying them and discovering more. So you have resistance to this one. You'll have cross resistance to closely related ones, but there are ones that are not closely related. So there are others. I guess what I would say is I'm committed to sustainability. So if you have something that's great, make it last 20 years or 50 years instead of like 6 months or 3 years. That's kind of where I'm at. That's what I was trying to say in the quote that was, I don't know, misunderstood or purposely distorted. So I see kind of a balance there. Yeah, discovery is great. We should keep looking for the next thing, but we should optimize the use of what we have. And so for example, right now, there are places where even though there's a lot of money going into R&D, and companies are eager to find the next protein or the next method of genetically engineering crops so they can control insects, there's going to be a gap of like 5 to 10 years in some places where there's no useful transgenic crop to control a particular insect. These are major pests. I didn't talk about it today. So for example, in India, the pink bollworm is resistant to the 2 BT toxins that are different enough that they don't cause cross resistance. The only 2 that have been genetically engineered in cotton that work against it, and there's like a 5 to 10 year gap with no new product in sight.
So, someone gifted you 500 acres of fertile cotton-growing land in India and said in 30 years you must use the best genetics and the best management strategies for insect-resistant cotton. That's what would you do?
So I guess the key would be integrated pest management. So think of the whole crop system and how the insect relates to that system, and use all the different tools available, like the timing of planting and harvesting, the spatial configuration, the extent to which the natural enemies are available to control the insect, whatever insecticides are available. And at the moment, the commercial BT cotton doesn't work against at least pink bollworm in some areas. But if I had either a new product that worked, or I was in an area where the pink bollworm, one of the main pests, was still susceptible, then I'd be really careful about how I used that. I wouldn't plant it 100% of my 500 acres every year. I'd probably do something different than the conventional refuge strategy. Convention is a refuge in space. I would use a refuge in time probably, because that allows for selection to act against resistance, and it also works independently of the movement patterns of the insect and the dominance and all that. If you plant, let's say if you can afford it in terms of expectations of pest damage, maybe it's every 3rd year you don't plant BT cotton at all. And in addition to that, I wouldn't plant, if I had those 500 acres, I wouldn't plant cotton every year. I'd plant something else that pest won't survive on. So it's kind of mostly it's going back to principles that were well established before there were any transgenic crops.
When I went to the Monsanto mixer at the conference, most of the Monsanto scientists were from USDA and other universities. And when I visit different universities, there were the ex-Monsanto scientists. The current debate, argument, those type of things may give them a temporary benefit, but I don't see that any benefit gained by actually people use crop by looking at those debate or arguments. So is there any way to just make them declare like their career path more clearly so people do not get confused by change of their position over time?
So you're saying that people are not clear about where their history is, whether they.
How
So
their opinion evolve, go back and forth.
So I should clarify, I guess, when I was asked before, these Monsanto scientists, why are they looking at one way and I'm looking at it so differently? So when the criticism comes from industry scientists who have a clear conflict, a clear vested interest, or at least they think they do, in looking at things a certain way, it doesn't bother me so much. What bothered me, the list of those people that I showed you are all university scientists. And I think we have an obligation, we're paid by the public, we have an obligation to at least try to be honest brokers, not to be tools of one side or another of vested people with vested interests that don't align with the public. So whether you be, you may be pro-GMO or anti-GMO, but as a university professor, I think you have an obligation to try and be as objective as you can and be as useful to the public if you can.
And with that, let's thank Bruce. I just want to note that this is an excellent example of the kind of discussion